So, after discussing into the details of composition of plasma membrane, now we will go into the today's session that how minerals are acquired by the plants from the uh, across the cell membrane, and how mineral could cross the cell membrane. So, how solute cross the plasma membrane via passive or active transport, and then we will explore the mechanism of various transport processes mediated by wide range of transport proteins and how these proteins are distributed for maintaining the right balance of ions within the cells. So, this is very interesting to go through in details. So, I hope that you will enjoy that how uh, these ions, these solutes move across the plasma membrane and what are the modes adopted by for movement of solutes. So, these are the basic content we will discuss in detail, that just a brief sketch. Now, you see that just I mentioned that transport of solute could occur across the plasma membrane by two modes. One is passive, another one is active method. So, you see that passive process and active process, as the name itself indicate, that passive process does not require any sort of expenditure of energy, whereas active process requires an input of energy. And moreover, you see that in terms of Gibbs energy, free energy change, generally in case of passive transport it is negative, whereas in active transport it is positive. And passive transport process generally is very much thermodynamically favorable, whereas active process is not thermodynamically favorable. Generally in passive process you see that movement of molecule could occur along the concentration gradient. So, you see that this statement is very important. Movement of molecule could occur along the concentration gradient, not against the concentration gradient. But in active transport, then movement of molecule could occur against the concentration gradient. You see that this passive transport if generally occurs along the concentration gradient, so it is a spontaneous process, and whereas active transport is non-spontaneous and energy is required in different forms, ATP, electron flow, proton gradient. So, likewise, just we will go into detail the sketch of these passive and active transport through this slide. You see that passive transport could occur via diffusion or facilitated diffusion. So, if you have a sketch in mind that how transport of solute could occur across the plasma membrane, just I mentioned it could be through the passive transport or active transport. Then you see that if it is passive transport, it could be accompanied by diffusion or facilitated diffusion. So, you see in the facilitated diffusion that diffusion is facilitated by something else. So, it could be facilitated through ion channels or transporter or carriers. Then if it is through the ion channels, it could be gated or non-gated, and if it is gated, then it could be ligand mediated, voltage mediated or mechano gated ion channels. Now, if it is transport is occurring against the concentration gradient, now it is known as active transport, and then primary active transport could take place for movement of solute or secondary active transport. Then this primary active transport is generally accompanied by various pumps. So, just to name a few: P-type ATPases, F-type ATPases, V-type ATPases, ABC and proton pyrophosphatase transport pumps. Now, if it is secondary active transport, I will discuss in detail these primary active transport, but just you must have in your mind the sketch of whole transport system. Then we will go one by one. That is also very interesting to share with you. Now, what is passive transport? Just I mentioned that it occurs along the concentration gradient, you can say downhill, without expenditure of metabolic energy. Generally, when the solute is uncharged, generally its movement is solely affected by its concentration gradient, and the combination of these gradients is generally termed as electrochemical gradient, which determines the direction of transport of charged molecules across the membrane. Generally, just I mentioned in the sketch of the passive transport, it could be simple or facilitated diffusion. So, what happens in simple diffusion generally solutes dissolve directly in lipid bilayer, just oxygen and carbon dioxide, and then diffuse across the plasma membrane without the involvement of membrane protein. And the direction of transport in simple diffusion is solely determined by the concentration gradient of the solute that exists inside and outside of the cell. You see in this figure that here it is cytosol and here it is extracellular fluid. So, movement of water, carbon dioxide and oxygen could occur across the concentration gradient. But you see that the rate of diffusion across the lipid bilayer depends upon various factors, uh, namely concentration gradient, the hydrophobicity and size of the solute moving across the plasma membrane. So, these factors determine that how various solute move across the plasma membrane by simple diffusion. So, generally simple diffusion through the lipid bilayer is not selective. That means it allows any nonpolar molecule that can dissolve in the lipid bilayer and then evenly distribute between the inside and outside of the cell. And just I mentioned the gases like oxygen, carbon dioxide and nonpolar molecules such as benzene, as well as small polar but uncharged molecules like water and ethanol could easily cross the plasma membrane. So, here it is very important to mention that diffusion and osmosis differ from each other. Osmosis is basically is a special type of diffusion where solvent is passively transported through semi-permeable membrane from an area of higher solvent concentration to an area of lower solvent concentration. So, you must keep in mind what is there is a subtle difference between osmosis and diffusion and osmosis. One require biological membrane and one do not require. Now, you see that we come to the after simple diffusion, what is facilitated diffusion, that we have just gone through that, like carbon dioxide and oxygen have ability to permeate through the plasma membrane directly, whereas others require assistance of to traverse the plasma membrane to the inside of the plasma membrane. Basically, this facilitated diffusion involves the movement of molecule across the plasma membrane with the aid of membrane protein, especially channels and carriers. You see, this is channels and carriers. They aid basically this facilitated diffusion, and generally the rate of solute transport across the membrane is significantly higher in facilitated diffusion as compared to simple diffusion. One thing you must note at this point, that the rate of facilitated diffusion is generally higher than simple diffusion. And this facilitated diffusion basically enables the passage of polar molecules and charged molecules such as monosaccharides, amino acids, nucleosides and ions across the plasma membrane. So, generally a concentration gradient exists for these molecules, so they have the potential to diffuse into or out of the cell by moving down. However, due to charge or polar nature, they cannot pass through the phospholipid portion of the membrane without the aid of these carriers and ions and channels. Hence, you see that facilitated diffusion, the term itself indicate basically transport proteins acts as basically shield and guiding these molecules to pass through the hydrophobic core of the membrane and then aid in movement of these solutes. So, this is facilitated diffusion. Now, what are the different proteins involved in mediating facilitated diffusion? So, basically you see that these channel proteins, basically they create open pores through the membrane and generally making unrestricted or free diffusion of molecules that fit their size and charge criteria. And transport through channels is always passive. One thing you must note, that transport of solute through these channels is always passive, and generally the rate of diffusion facilitated by channel protein is typically faster than that facilitated by carrier proteins that I will discuss in my coming part of the lecture. And these channels generally exhibit less specificity in terms of the shape of the molecule they transport compared to carriers, and they often do not have a maximum capacity for transporting molecules unlike carriers. So, basically channels that specifically facilitate the transport of inorganic ions, they are basically referred to as ion channels. You must note very important point. So, basically ion channels, they show high selectivity for certain ions, and most of the ion channels are generally not continuously open. Instead they can be regulated to open or close under different conditions. So, channel proteins contain a particular region called gates that open and close pore in response to signals, like some channels known as voltage gated channels, just I mentioned in the first sketch of solute transport. And these voltage gated channels open in response to changes in electrical potential across the membrane, such as sodium channels, potassium channels and calcium channels, while other is known as ligand gated channel, just I mentioned. And these ligand gated channel, they opens in response to binding of specific signaling molecules called ligands. So, this one of the best example of ligand gated channels are acetylcholine receptors. Now, you see that this is the very important technique which help to identify these ion channels, particularly in guard cells, and reveal the crucial role in regulating the solute movement during the opening and closing of stomata. And these patch clamp technique has helped to recognize that ion channels are present in all plant cells located in both plasma membrane and tonoplast, and these ion channels are investigated using a very crucial technique known as electrophysiological technique, such as patch clamp technique. So, you see in the figure, this technique basically involves gentle pressing a blunt tipped glass, you see blunt tip glass micropipette known as microelectrodes, you see here, against biological membrane to basically measure extremely small electrical currents, typically in very low range. And this patch clamp technique help to determine the minute electrical currents generated as ions pass through the channels. So, one interesting aspect of this patch clamp technique is to ability to observe the activity of individual protein molecules, particularly channels, as they facilitate the movement of ions. So, this technique is very much helpful, that how ions move and the minute current generated that can be observed by this patch clamp technique. Now, what are the different types of ion channels that are present? We will discuss that. Basically, important cation channels found in the plasma membrane are potassium selective channels and basically these potassium selective channels can transport potassium ion, both inward and outward. But as you see that calcium selective channels generally permit the passage of a range of anions like chloride, nitrate and organic acids. So see the difference that these cation channels, what they are meant for. Like one discussed potassium selective channels and calcium selective channels. So basically the calcium selective channels, they are specific anion channels uh, generally dedicated to organic acids like malate. And channels basically have a tight could be regulated through conformational changes that basically act as a sort of switch or control between the permeable or non-permeable states of these channels. So the transition between open and closed states known as gating, gate are open, gate are closed, is generally this opening and closing of channels, or you can say gating, is basically controlled by either voltage or ligand chemical that bind to these channel proteins such as hormones, calcium ions, G proteins and pH. So some channels they are sensitive to stretching and response to change in cell target pressure. You see that guard cells responsible for regulating stomatal opening and closing possess at least 4 types of calcium channels. One is regulated by voltage, another by calcium ion, and remaining 2 by regulated by ligands. So this is very important point. And then various types of cation channels they are also present, and namely one is potassium channel, and these potassium generally channels are involved in potassium flux across the plasma membrane of the guard cell, and generally these potassium channels they aid in potassium uptake from the soil and facilitate the release of potassium from living stellar cells to xylem vessels and contribute to potassium uptake in different parts of the plant. Now this is the slide showing the how potential, what are the different types of potassium channel exist. So generally these potassium ion channels permit transport of ion only in one direction. So distinct categories of ion channels facilitate inward and outward currents, with channels exhibiting rectification responsible for this one-way flow. For example, in this potassium inward rectifying channels and potassium outward rectifier, initially they were identified in guard cells, and but later on these channels, inward and outward rectifying channels, were identified in various plant cell types. Basically inward rectifying potassium channels, also known as potassium influx channels, are activated by membrane hyperpolarization, and activation of these channels in plant cell occur with an increase in cytosolic calcium concentration and triggering potassium activity of potassium efflux channels. Now channel is responsible for calcium transport, and the mode of basically calcium primary mode of calcium intake into the cytoplasm of eukaryotic cells occur through the ion channels rather than you see that proton coupled transporters. So you see that generally the concentration of calcium in the cytoplasm is very low, approximately about 10 to the power minus 7 m, whereas the concentration of calcium in soil or apoplast and vacuoles generally ranges from 0.1 to 1 millimole. So you see that how the concentration of calcium vary in the cytosol and in vacuoles and soil. So basically this difference in the concentration of calcium creates calcium gradient between the external environment, that is soil, cell wall or vacuole, and the cytosol, which allows calcium channels associated with the plasma membrane and tonoplast to efficiently transport calcium ion into the cytosol. Basically, plant calcium channels typically exhibit permeability to other cations in addition to calcium, and generally a steep calcium gradient uh, basically enables non-selective calcium permeable channels to induce a rapid increase in cytoplasmic calcium levels once activated. Among various plant calcium channels, one of the primary calcium channels located in the plasma membrane of the guard cell is generally triggered by hyperpolarizing potential and also triggered by abscisic acid and reactive oxygen species. So, in addition to this, plant cells, you see that significantly number of inorganic ions like nitrate, malate is present inside the cytosol. So, basically anion channels basically create energy gradients that generally facilitate passive flux. Initially, these anion channels were identified in guard cells of the plant cell, and these channels such as calcium activated R type and slower activating S type responds to voltage change, resulting in the expulsion of colloid ions and for depolarization of membranes. So, basically this depolarization of the membrane uh, triggers the activation of outward rectifying potassium channels, subsequently contributing to salt loss and reducing the plant turgor. Now, in addition to this, you see that carrier proteins known as transporters or permeases. So, unlike channels, these transmembrane transporter or proteins do not create pores in the membrane. So, what is the way out? What is the other you say contingency plan, you can say? So, these carrier proteins, they selectively bind non-covalently with solute at very specific site, and then inducing a sort of conformational change in carrier protein that exposes the solute to other side of the membrane. You see, they bind and then bring about the conformational change, you see, and then once they bind and they bring about conformational change in the protein and then in these carrier proteins, and then reaches to another side of the, or you can say in the cytosol. So, you see that then once solute reaches to the cytosol, it is released from the binding site, and then carrier protein returns to the original conformation, and then it is further ready to transport another solute molecule or ion. So, this process of binding and release generally resembles an enzyme catalyzed reaction. So, it is very interesting to note. So, this binding of solute to the carrier proteins resembles the enzyme catalyzed reaction, and carrier mediated transport allows for the transportation of a range of solutes, but generally the rate of operation of this movement is very slow. It could range to 10 to the power 4 to 10 to the power 5 molecules per second, and compared to channel mediated solute transport. So, this is a very interesting point to note. Now, come to the active transport. We have discussed in detail the passive transport. Now, active transport is the process that generally moves ions or molecules against their concentration gradients, and it could be mentioned that active transport is meant for movement of solute from low concentration to an area of high concentration, or you can say against the concentration gradient. So, this basically this movement is facilitated by carrier proteins. These carrier proteins, you see, they are embedded within the cell membrane. So, unlike passive transport which relies on the natural flow of molecules down their concentration gradient. Active transport basically requires energy from the cellular metabolism uh, to carry out this process, and basically this energy is derived from breakdown of ATP, and which serves as energy currency in the cell. So you just see that active transport leads to accumulation of solutes on one side of the membrane and then creating a sort of concentration gradient. But transport of solute occurs along the electrochemical potential gradient and does not require expenditure of energy. But if it is occurring against the concentration gradient, it requires an input of energy. That's why name active transport. Now, active transport could be categorized into two main types. One is primary active transport, another one is secondary active transport. So first we will discuss in detail what is primary active transport, and this primary active transport is directly coupled with metabolic energy source such as hydrolysis of ATP, and generally ATP is broken down into ATP and inorganic phosphate. And in some cases what happens, primary active transport can also be driven by absorption of light by certain carrier proteins that has been observed in certain bacteria. And carrier proteins involved in basically in this primary active transport, or they are basically referred to as pumps, because they actively pump ions or molecules across the plasma membrane using energy derived from ATP or absorption of light. And majority of these pumps facilitate the transport of ions such as proton and calcium across the membrane, and they are categorized as ion pumps. And some pumps, those classified under ABC transporter, are basically capable of transporting larger organic molecule, organic solutes across the membrane. So basically ion pumps can be classified into two types, namely electroneutral pumps and electrogenic pumps. So you see that this one is electroneutral pump and this one is electrogenic pump. So first, what are the electroneutral pump that transport ions without resulting in a net movement of charge across the membrane? So basically charge does not change. For example, potassium ATPase generally found in certain animal cells expel one proton and for each potassium ion taken in, and then maintaining the electroneutrality. So you see that charge is balanced in this case, but opposite to it in electrogenic pumps, generally the transport ions while generating a net movement of charge across the membrane. For example, proton ATPases found in both plant and animal cell expels proton ions resulting in a net positive charge. Similarly, sodium potassium ATPase in animal cells like neurons is electrogenic, and as it expels 3 sodium ions for every 2 potassium ion taken in, and resulting in a net outward movement of 1 positive charge. So basically in plant cell, most common electrogenic pumps are proton ATPase. This you just note down the example of electrogenic pump that exist in plant. These are basically proton ATPase, proton pyrophosphatase and calcium ATPase, and basically all these electrogenic pumps transport ions outward. And in addition to this, there is secondary transport also exists, and the secondary transport are necessary for uptake of other mineral elements. Now first I will discuss what are the proton ATPase pumps. These pumps are referred as p-type ATPases and present in the plasma membrane, you see, of the tonoplast and other cellular membranes. They are basically structurally different from F-type ATPases, which typically found in the mitochondria and chloroplast, such as ATPases synthesize ATP. However, proton ATPases operate in reverse manner by hydrolyzing ATP. You see here ATP is hydrolyzed. So, you see in this figure a model of this proton ATPases have been depicted. You see that this type of proton ATPases, they are also known as p-type ATPases, and sorry. So, in this figure generally the enzyme protein consists of a single polypeptide chain figuring 10 hydrophobic transmembrane segments or domains. You see they are named as 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, and they have 10 hydrophobic transmembrane segments or domains, you can say, and only out of these 10, only 3 of these segments are helical in nature, namely 4, 5 and 6, and while others they are present in the form of cylinder. So, these segments are basically interconnected by hydrophilic loops that extend into the cytosol and the cell wall, and generally ATP binding site is believed to be located at the aspartic acid residues that is position on the loop connecting the 4th and 5th segment toward the cytosolic side. So, once ATP hydrolyzed, the protein undergoes a sort of conformational change, facilitating the transport of 1 proton ions. You see transport of 1 proton ion from the cytosol to the outside across the plasma membrane. So, this is the basic structure of proton ATPases and how it works, how they are made up of. It is very much clear from this slide. Now, vacuolar ATPases. Vacuolar ATPases are basically proton pumps that are basically electrogenic, means they transport protons from the cytosol to the vacuole and generally helpful in establishing a proton motive force across the tonoplast. So, these basically electrogenic proton pump basically creates sort of pumping in the vacuole, and generally which is in the range of 20 to 30 millivolt, and generally these vacuolar ATPases play a very important role, particularly to maintain the bulk electrical neutrality. Anions such as chloride or malate ions are transported from cytosol into vacuole through channels present in the tonoplast. So, this process of basically anion transport ensures the conservation of bulk electrical neutrality and enables the vacuolar proton ATPases to generate a significant concentration gradient of protons across the tonoplast. Now, these are the differentiating points that exist between the plasma membrane proton ATPase and vacuolar ATPase and how they differ from each other. That you see that the proton ATPases found in the plasma membrane and tonoplast facilitate basically they exhibit differences, and you see that plasma membrane proton ATPases differ from the vacuolar proton ATPases. You see these plasma membrane proton ATPases are typically inhibited by 1-date ions, you see here, and but these vacuolar proton ATPases, they are insensitive to 1-date ion, and these plasma membrane proton ATPases, they are not sensitive to nitrate, but they are inhibited by nitrate, and this plasma membrane localized proton ATPases transport single proton per ATP molecule hydrolyze, whereas you see that 2 protons are transported per ATP molecule. So this is a very significant difference between these two types of proton ATPases. Now come to the proton pyrophosphatases. These pumps are located in the tonoplast. Although these proton pyrophosphatases could also occur in the membranes of Golgi bodies. And basically these proton pyrophosphatases serves for pumping of protons into the lumen of vacuole and Golgi cisternae, and these pumps are believed to work alongside the vacuolar ATPases to establish a sort of proton gradient across the tonoplast, and vacuolar proton pyrophosphatases transport only one proton per pyrophosphate molecule hydrolyzed. So these are the different types of ATPases found in different organelles: P type, F type, V type, and proton pyrophosphatases. So we have discussed individually these different types of ATPases pumps, and you can see the difference existing among each other. So you must be very clear how they differ from each other by seeing this slide. Now calcium pumping ATPases, basically they are also playing very crucial role and present in the plasma membrane, tonoplast, and other membranes like chloroplast and endoplasmic reticulum. So basically they facilitate the translocation of calcium and across the membrane by coupling hydrolysis of ATP. In the case of plasma membrane calcium ATPase, calcium ions are basically pumped out of the cytosol, and this action basically helps, or you can say aid, in maintaining the low cytosolic concentration of calcium to prevent precipitation of phosphatases and to keep calcium dependent signaling pathway operative properly. Now ATP kind binding cassette transporters are responsible for transporting specific large metabolites such as anthocyanin and other secondary plant products from the cytosol into vacuole by traversing the tonoplast, and these transporters basically situated on the tonoplast, which directly utilize the ATP for their proper functioning. More recently you see that this ABC transporters have also been identified in the plasma membrane and also in the mitochondria, and most ABC transporter function as sort of pumps. Some members of their superfamily serves as ion channels that undergo opening and closing facilitated by hydrolysis of ATP. Now secondary active transport is very interesting. We will go in details that how nutrients are transported across the cell membrane against their chemical or electrochemical potential gradient through a mechanism known as secondary transport. So basically this process, secondary active transport, does not directly utilize the energy released by ATP hydrolysis, but rather than indirectly generate the energy stored in the proton electrochemical potential gradients across the membrane, which is also referred as proton motive force. So the electrochemical proton ATPases pumps act as a proton translocating carrier proteins, translocating carrier proteins, which serves conserving the free energy from hydrolysis of ATP in the form of proton gradients across the membrane, with more protons accumulating on the outer side. So this proton gradient basically combined with the typical membrane potential contributes to proton electrochemical potential gradient, or you can say proton motive force, which generally tends to move proton back across the plasma membrane through specific carrier proteins located elsewhere on the same membrane. And when the proton returns to the cytosolic site, the proton motive force generated by electrogenic proton transport can be utilized to facilitate the transport of other solute molecules or ions against their chemical or electrochemical potential gradient through the same carrier protein through which proton is returning to the cytosol. So this phenomena is basically known as secondary transport because ions or molecules of the two different substances are transported simultaneously through the same carrier protein, and this process, the solute is of transport, is regarded as co-transport. Now, secondary active transport could occur in two main types, symport and antiport. And you see that in symport, influx of photons is generally coupled with the movement of another solute in the same direction. You see, symport transport is occurring in the same direction, and here in antiport transport is occurring in the opposite direction. So, you see that generally in symport the influx of photon is coupled with the movement of other solute in the same direction. So, energy is in symport, energy is basically dissipated by a photon moving back into the cell, was generally coupled to the uptake of one molecule of substrate, for example sugar, into the cell, and the carried protein involved is known as symporter protein. This is symporter protein. Then opposite to it, how antiports work. In antiport, the influx of photon is generally coupled with the efflux of another solute. So, here the energy is dissipated by a photon moving back into the cell, which coupled to the active transport of substance, for example sodium ion, out of the cell. So, this co-transport mechanism, you see that uh, is termed as antiport, and the carrier protein involved is called antiporter protein. In both cases, you see that substrate is moving against its concentration, again its gradient of electrochemical potential. Both neutral and charged substrate can be transported by such secondary active transport. Now, here you see this overview of various solute transport processes that localize in the plasma membrane and toloplast, you see from this slide. Now, you see that in summary that how solute moves through the lipid bilayer of the membrane, that could move freely or by simple diffusion, or it could be facilitated diffusion. Then how primary active transport uh, then helps to movement of various solutes, and then secondary active transport I have discussed in detail, that is meant through symporter and antiporter. And then all these aspects we have covered in detail. Just in a summary, we have just mentioned the important points in a nutshell, and how various pumps are involved in transporting solutes. So, we have gone into in detail all these aspects. So, this is all about the transport of solute across the plasma membrane, and we have discussed in detail passive transport and active transport. Thank you very much for patient listening, and hope you might have enjoyed my lecture. Thank you very much.